So last time we had just finished talking about transcriptional regulators, right? And we had talked about how you just need a few regulators to regulate how, all the genome. Um, Comparatively speaking, yes, there's still lots and lots of them, but comparatively speaking, there are relatively few regulators that can multitask and affect multiple places on the same genome and regulate the gene expression related to it. And then we had started talking about differentiation and talked about how cells become from that totipotent original cell to the pluripotent cells, which are partially differentiated. And then you have those, specific stem cells that are related to each organ system, like your meioblast or neuroblast. And that process of differentiation, again, just takes just a few regulators to get you to all the different cell types that you see inside the body. And the way that happens is through very regulated, timed expression, selective expression of these proteins in specific subsets of the stem cells. So if we go up here, you had seen that, right? That you have a small subset. Once the polarity is decided inside your embryo, once you have a head and a tail end of that embryo, once that process has started, as you might have learned in that self-assembly in the genome as well, right? That's where a lot of that comes in as well. We see that in specific sections of the embryo, you start to get specific regulatory proteins, expressed that then determine what cell types are going to come out of it and how these cells are going to differentiate. So within a few easy steps, with just, 3 main proteins, you can start to now make many, many, many different types of cells, differentiating from that original cell. So this is how we do stem cell research as well. We can actually take fibroblasts from cells, and we can specifically introduce these regulators, these master cell differentiation regulators, and we can generate IPS, a subpopulation of what we call induced pluripotent stem cells. These are stem cells these are fibroblasts that have de-differentiated. So they've gone backwards in time, kind of, and become into a little bit more pluripotent cell stage, where they can then be induced in culture to make specific types of cells, like muscle cells or neurons or fat cells, skin cells, whatever it is that you need. And that's one very, very, very hot part of research, is to develop these type of systems that can help us kind of combat the need for transplant organs, right? Is to either find cells that can be then introduced into an actual body, or make whole organ systems that can then be used in place of transplanting organs from an individual. So now we are going to shift our gears and we're going to start talking about post-transcriptional controls. Everything we talked about last class was transcriptional control, or pre-transcriptional control as far as gene regulation was concerned. So it was before we actually get to an actual physical transcript or a copy of that gene that we are trying to make. So in post-transcriptional control, the first set of controls come at RNA splicing, that messenger RNA processing, something that we have talked about before in detail. That's the whole process through which our mRNA gets organized, especially in eukaryotic genomes, right, where you have the intronic and the exon sequences, where those introns get spliced out. They can be differentially spliced to put together different isoforms of the same protein in different environments. You can regulate how long this mRNA is going to be around based on the poly A tail length that can affect its stability and its half-life essentially inside the cell. We can regulate how much of this mRNA is going to be translated, or at what rate it is going to be translated, based on other 5' and 3' untranslated region information that's there, that's regulatory information for this RNA. There are other types of things that also happen, right? And then you have those bacterial genomes, right? Where you have those multi protein or multi recipe transcripts where a single promoter is regulating a whole set of genes, right? In a particular system. And that again can be spliced, not spliced, but that is translated in different ways. Now, also in bacteria, you can have regulation of how much the transcript is going to be translated, similar to eukaryotes, by regulating the ribosomal binding site. So the place on the messenger RNA where the ribosome is initially going to come, that is a particular short sequence that the ribosome is recognizing in bacteria. And that can be bound with the inhibitor or change its conformation under certain conditions through, because mRNA is a single-stranded molecule, it can cause those tertiary structures, right? And those structures, the secondary and tertiary structures, can inhibit binding of the ribosome, so it is not translated under certain conditions. So even though you have a single cell organism in the case of bacteria, you can still have sophisticated control of the messenger RNA and still have those controls to make sure that you're not translating protein when it's not needed. Another thing that we have is the small regulatory RNAs. These are those microRNAs and silencing RNAs like the ones that I've shown you in that cartoon video about RNA interference. Those are typically going to inhibit expression of the RNAs that are related to them, usually complementary sequences to those mRNAs. So microRNAs bind to complementary sequences in messenger RNA, and by doing so, they can make these little double-stranded areas that essentially act like blocks and control expression of the gene, of that particular, translation of that particular gene. And then the RNA interference is where you have a foreign RNA that interfere with the expression of a known messenger RNA that's there, the gene that's there. That was the example with the one that we looked at, where the purple gene was foreign, was introduced from outside. However, the way it was introduced caused that double-stranded structure, that mirror image recipe that caused it to be then all destroyed instead of kept there, right? So going back to bacterial gene expression and how that can be controlled, this is just an example of what we were just talking about, that just like eukaryotic mRNA, the bacterial mRNA is also going to contain areas that the ribosome is recognizing. Now, there are specific sequences that ribosomes recognize and binds to that is very close to the start site, which again is the AUG, which is your methionine. And right before that, usually what you have in bacterial messenger RNA is a particular pin loop structure that kind of signals the ribosome, or kind of, finds that ribosome, targets the ribosome. It's like a little light bulb going off, so the ribosome can recognize and bind next to it. Well, in certain cases, a repressor or interfering protein can bind to that pin loop structure, so that now the binding site is blocked and the ribosome cannot come in and bind to that AUG start codon and cannot make any protein. So in this case, while the mRNA is present, if we were to look at this particular messenger RNA in an experiment, we'll be like, well, it's expressed, it's there, but it won't be translated, so there'll be no functional protein. So it's being made, but it's not being translated. So how do these things happen is usually because of environmental sensors, or just, either internal environmental sensors or external. So there's one place where, one simple example is just increasing a temperature. Increasing the temperature will cause this pin loop structure to get opened up. Sometimes this is an example where the pin loop structure contained the start site within it, and it was blocked because the pin loop structure was there, the secondary structure was there. However, if you increase the temperature, these binding sites can open up. And now the AUG start codon can become available to ribosome to bind and make protein. Now, this can definitely go the other way as well. So if you think about in this case, where AUG was not part of the pin loop structure, destroying the pin loop structure will actually inhibit the ribosome from binding, right? So if you are looking at a question, look at it carefully to see if the AUG codon or the start of your actual mRNA, the protein, is in the pin loop structure or outside of it. Because if it's within it, then heating it is going to expose it and it can bind to it. If it is not part of it, then it may actually do the opposite. So two ways that bacterial genomes are getting regulated. One is through repressors on the messenger RNA to stop the translation. And the second is environmental stresses like heat or cold that can disrupt those secondary structures that were acting as a way to either prevent or enhance translation. And that can then have the effect, the appropriate effect on them. Okay? Okay, so then you have your microRNAs. MicroRNAs can actually occur quite naturally as a regulatory mechanism inside our cells. When you have a lot of a particular messenger RNA present, especially for housekeeping genes, you can get to a point where there's too much of it present inside the nucleus. At that point, there is only going to be certain, there's a certain threshold, there's certain level of processing that's happening of that messenger RNA as it's getting transcribed. All that extra mRNA that's transcribed ends up making these little looped secondary structures, double-stranded structures, where you have areas that are loops and areas that are double bonded. This precursor mRNA, in this case, is not your final messenger RNA like it would be in a normal condition when it is processed, fully processed appropriately. Well, this double-stranded structure is not recognized by the nuclear mechanisms of processing, so it gets exported to the cytoplasm the way it is. And once in the cytosol, these structures can get chopped up into smaller double-stranded pieces through proteins called RISC proteins that will then create smaller pieces of this very long messenger, pre-processed messenger RNA. These pieces can bind to the messenger RNAs that are already processed, that are already in the cytosol, because they're complementary to it and they can bind to it, just like they did before. And when they bind to them, that inhibits the translation and it targets them for degradation. That's one way that we target a messenger RNA for degradation, because there's too much of it around essentially. So what it is doing is that the precursor messenger RNA, or the microRNA in this case, is going to be, the precursor microRNA is going to be processed through the RISC proteins to make single-stranded microRNA by chewing all these pieces up and making those single strands that will be complementary to their target messenger RNA. They'll bind to those and mark them essentially for degradation. Think of it like the ubiquitination in proteins, how proteins get ubiquitinated and get targeted for degradation. In this case, the messenger RNA, the fully processed messenger RNA, when it finds that complementary piece, gets targeted for degradation. Now, the amount of this microRNA that binds to those messenger RNA will regulate how much of it is going, how quickly it's degraded, right? So if there's a lot of it getting bound, lots and lots of microRNA pieces getting bound, and they are all, there's extensive match in the complementary structure, it's going to be rapidly degraded by the nuclease that are also part of the RISC complex. In the other case, however, if there's just portions of it that are matching and not the entire sequence, so you still have some single and strand tails, in that case, it will still inhibit translation. So it's still not going to be translating as efficiently as it used to at the same rate. But this time it's going to be kind of just getting slow translation and then eventually getting degraded by the RISC nucleases. So the same thing will eventually happen, but it will be at a slower rate. It's just going to slowly diminish the amount. Now, silencing RNA or RNA interfering RNA kind of works the same way. In this case, we introduce, or in some cases, a bacteria may introduce this inside a human cell or its host cell. You get a foreign double-stranded RNA piece that's going to be similarly digested and cleaved as the RISC proteins. But what this is doing is that it's taking that foreign double-stranded RNA and it's chopping it up into smaller double-stranded silencing RNAs, usually about 20 nucleotides long. This process is done by dicer, essentially dices it up, right? And then the RISC protein, again, do the same type of process as they did before. They treat these just like microRNAs, where they split them apart and you get single-stranded silencing RNAs that can bind to their complementary messenger RNA and target that, again, the same exact way for RISC-related nucleases to come chop them up and destroy. Okay? So this is one way that our body also actually has its own silencing RNA system to destroy foreign RNA as well, to protect us from infections. When such double-stranded RNA, foreign pieces of RNA are seen, they are usually recognized automatically as infectious. And that's why anything related to that is destroyed. Okay?
That's super cool.
Isn't it? I know.
That's really cool.
We use it to destroy their own proteins, but you know, it's naturally a defense mechanism by the cells. So another thing that the silencing RNA can do is actually go back to the DNA and silence the actual it can do transcriptional silencing as well. And this is again another way that our body, this is a defense mechanism by our body to especially keep those transposable elements, those jumping genes in check, so that they are not getting transcribed or moved or shifted around. That's done through a very different sets of proteins. So instead of RISC protein, these are RITS proteins that are transcriptional silencing proteins. And what they do is, again, the same way, they will bind to these silencing RNAs. They will open them up so you have single-stranded siRNA that can then search for any RNA that's complementary to it inside the DNA, inside the nucleus as it is getting transcribed, and essentially target that for degradation, but also target that portion of the genome for histone methylation so that you get heterochromatin there and you don't do this process again. So it kind of checks all those new transposable pieces that kind of come in and out to make sure that they are not transcribed and are not causing a problem. Finally, there's a longer version of these non-coding RNAs that can also have many different types of functions inside the cell. Now, these long non-coding RNAs are going to be those RNA that have functions, actual physical function inside the body without being translated into proteins, right? So ribosomal proteins are, ribosomal RNAs are kind of long non-coding RNAs, right? They serve other functions inside the cell without getting translated into a physical protein. Usually these work in combination with a protein partner to bind to make the formal quaternary structure, right? And these can interact both with RNA as well as DNA, depending on what they're doing, right? Whether it's a tRNA or a ribosomal RNA, they're going to be interacting with both the DNA. Some are going to interact with the DNA, some will act with the RNA. Or part of DNA polymerase, for example, any one of those will have these RNA components. So any questions on this, the transcriptional regulation before we go to cell signaling? You guys are good? Yes, no?
Okay, so for this last slide, you just said that there were two proteins and they are the ones that help for the regulation of the both of
No. What I'm talking about is that there are many RNAs inside our body that have different types of functions, right? Other than making proteins. And usually some of them may work by themselves, but many times these are going to be part of a bigger structure. Like think about in the case of ribosomes, right? It's not just the ribosomes, ribosomal RNA that's important. It's the ribosomal RNA with its protein complex. Together, they will work to do whatever work they need to do, right? There are also long coding RNAs that are recognized by specific proteins and that then take them, localize them to specific area, either on the DNA or on the RNA to do transcriptional regulation in their own way. That's also something that can happen. They are part of the tRNA is part of that translation machinery, right? That's going to bring your codons and with its anticodon. And it's an important part of working machinery inside the cell. So long non-coding RNAs, I'm here kind of lumping two different types of RNAs together, right? These are all functional RNAs that have actual physical function inside the cell without getting translated into a protein product. Yes? So it's all those RNAs that are folded up into their own little tertiary structures that then do function inside the body. Did that make more sense?
Yes, Professor, thank you.
I have a question about the microRNA.
Yes.
Like you mentioned, it's synthesized inside the nucleus when there's too many mRNAs in the cytosol.
So there, again, that's a simplistic version of it. There are sometimes environmental triggers that can cause it to be made as well. But one of the ways that microRNA is seen made is when there is too much transcription, but you don't need that much around. So when there is enough transcript kind of floating around and now you're still transcribing, it's an internal way to kind of do a feedback loop to control the transcription by creating these microRNA precursors that are complementary to that strand so that they can go and target them for degradation. And so if you look at this one, if I make it big again, 1 second. If you look at this, essentially what these are doing is they are going to those already processed messenger RNA. They've already gone through all the processing and everything and their final messenger RNA. They're binding to those and they're targeting them to get degraded or to have reduced translation to control how much final protein product is getting made by them. Okay?
Okay, got it. Thank you.
Okay. And one of the ways that these microRNA, what I was talking about, can occur is by the actual rate of the processing being constant inside that nucleus. So the as this transcript is getting transcribed, as the messenger RNA is getting transcribed, it's usually, if you remember, it's already getting processed. Well, there's only so much processing that can happen at a time. So all that extra transcript that's getting transcribed has chance now, instead of getting processed, to make those spin loop structures and make these kind of microRNA structures that can then be broken down into smaller pieces, which is this double-stranded RNA intermediate that is then processed by RISC that can then find its matches and target it for degradation. Okay?
Yes, thank you.
Okay, so now we're gonna go ahead and start our second set of information lecture, which is about cell signaling. Now, for cell signaling, please do remember that there is that link to another lecture that I have online. That's great as an introductory information as well. Has anyone watched it yet? This one from the MIT OpenCourseware. She does a great job at breaking it down into simple pieces and going over that. So I would highly suggest that you watch that as well. You may like her better than me, and then you can listen to her second lecture on this topic, and that will give you even more information. So here we go. We are going to talk about cell signaling. So the topic for today was cell signaling. And so we're probably going to do like basic overall cell signaling and talk about some main ways that our cells communicate with the outside world and through each other through this process. And then we'll leave the more specifics for later, probably to get through the end of it. So to start with, we're going to start by talking about general principles of cell signaling. We're then going to talk about G protein coupled receptors. And then we will start to talk about enzyme coupled receptors a little bit, but we'll probably focus more on the G protein and the general principles of cell signaling for today. Okay. So signal transduction is the way that we take in signals from the environment, and then we process them. Sometimes we amplify them if needed in order to have the appropriate response in order to survive or to do the works that we need to do inside our body, right? So you can think about it like a cell phone, as it points out in the book as well, where you're taking in the signal as one form, but you are responding in a very different way. So it's a radio signal in and sound out in that case. However, in the case of the cells, you will have some molecule that will act as the signaling molecule that will be bound to usually a receptor on the plasma membrane in the form of a protein or some other form. And that receptor then takes in that signal, changes somehow to move that information into the cell, where the intracellular then signaling molecule can go out and do the work that it needs to do. Okay. Okay. So the way we respond to these signals is essential for our survival at the end of the day, right? So there are multiple functions that we can do, obviously, in response to a signal that we receive. And this is just showing you different kind of responses at the end of the day, the big categories of responses that we can go through. So one way would be to feel the difference in the environment. Maybe it's the change in temperature, maybe it's change in pH or something else that tells us that we need to adapt somehow, change our internal environment in order to survive. We can also get signals from outside that help us figure out when we should be growing or dividing to replenish the supply of cells that is needed to perform a function. For example, in livers, if you have a lot of toxic exposure to a drug like acetaminophen or alcohol or something happens to it so that suddenly, 20% of the cells are dead or are stressed, there will be a signal that will be received by the remaining cells inside the liver, the hepatocytes in the liver, to signal them to divide, to replenish the supply of those cells that are about to get lost or that are lost. And those signals would be different than what you would have used otherwise in order to get your survival signal. Similarly, you can get signals that will induce the cells to differentiate into a particular type of cell. For example, your hematopoietic stem cells in the bone marrow, depending upon their daily signal that they are receiving from the environment, from the body, they will have different rate of production of red blood cells versus, neutrophils versus platelets, whatever it is that they need at that time, what our body needs at that time based on its current state. And then finally, there are cases where the cells are extremely damaged, something major has happened, or the environment has become unsuitable for it to survive. And then there will be specific signals that will target it for death. It could be through apoptosis or programmed cell death, or it could be something different, some other way to kill it. So now, how do we respond to these signals will be dependent upon what cell is receiving that signal and what environment it's present in, right? So the same signal can have a very different effect, very different response if it is targeting a heart cell versus if it is a salivary gland cell versus a skeletal muscle cell. And this just kind of shows you an example of that with acetylcholine, which is a common signaling molecule all throughout our body. And acetylcholine, if it is binding to a heart pacemaker cell, it's going to regulate your heart rate to be slower, very different response. However, in a salivary gland cell, it's going to cause it to secrete saliva. Again, a very different response through the same exact molecule neurotransmitter binding to its receptor protein, same receptor protein, but a different environment that it's going into. Here, it's looking at a heart pacemaker cell, which job is to do the heart rhythm, while in the second one, it's looking at a cell that its job is to secrete a mucus or a particular product. Similarly, in the case of the skeletal muscle cell, in this case, even the receptors different. There are different receptors that acetylcholine can bind to. So in this case, it will bind to the receptors that are located on the skeletal muscle cells and cause that cell to contract. Now, in some ways, yes, Taylor?
Does it matter the cell that's receiving it or the receptor, or is it both?
It's both. But that's what's important is that the same receptor on two different cells can have two different responses, two different types of cells, right?
Oh, okay, cool.
All the heart pacemaker cells will always behave the same way, but a heart pacemaker cell versus a salivary gland cell versus skeletal muscle cell, even the same receptor is going to have a different response based on the type of cell it's on. And obviously, if it's a different type of cell, a different type of receptor altogether, that would be a different signaling cascade anyway. Okay.
Awesome,
So it's a
thank
little
you.
bit of both. Both things need to be checked, whether it's the same type of cell or whether it's the same type of receptor or different receptor. Okay. Now, in some ways, when you think about it, two of these responses are very similar. Both the secretion response, yes, that's done through these little vesicles that are getting merged and secreted out, and the contraction, they are going to be very related to each other as a response, but at the same time, they're very different. So these signals don't have to be present right where they are affecting. Many be a primary response and the other two be a chemical response and secondary response. So we will talk about primary versus the secondary response later today and later in the class as well. It depends on how many steps in the process you're going to have and whether that particular signal came from right at that close by or far away. So just the fact that it is binding to this receptor and causing a process inside is not enough to figure out whether it's a chemical response or a secondary response. Okay.
Thank you.
Okay, so signals, especially neurotransmitters and many of our hormones, for example, they can act over a very long range of distance because they're able to just move through our bloodstream they can be made in one place, but then go all the way to another place, another organ system, and have an effect there. So these are 4 different ways that you can have signals interacting with their final destination. In the endocrine system, which is your typical way to secrete hormones, the endocrine cells are the ones that are secreting the hormones. These hormones usually have to go through the bloodstream to the target cells where they bind to the receptors on those target cells and elicit a response. In a paracrine signal, you have an intermediate kind of mediator in between the place where you have your, signal getting made and then the target cells. So here you have a signaling cell. This is, the main cell that is secreting the molecule that's going to act as a mediator. That signaling cell will secrete those mediators that will then, that signaling cell is going to be right where the other cells are. They're going to be very close by, right? And it's going to signal to the cells adjacent to it, essentially, cells that are nearby, right next to it, to have a response. So the signaling cell itself is not responding to this mediator. Only these cells that are the target cells are going to respond to that mediator. Then you can have one where it's a contact dependent signal, where the signaling cell has to be in physical contact with the target cell in order to respond. So in a paracrine signaling, it doesn't have to be, it has to be close by, but it doesn't have to be bound to the cell that it's affecting. But in contact dependent cell signaling, the signal never leaves the signaling cell. It's membrane bound. So it has to be in physical contact with the target cell in order to respond, right? So in paracrine, it's a secreted signal. In contact dependent, it's an integrated signal with the plasma membrane. And then finally, you have neurotransmitters and neuronal signals, which are going to be moving through the axons and then forming at that synapse with the target cell. They will elicit a response. Now, the neurotransmitter may pass the membrane or it may elicit the response another way. Okay. So we are going to kind of go through each one of these a little bit more in detail in the next few slides. So again, going back to the way these molecules interact with each other, we are first going to look at a molecule that is secreted. So this is a signaling molecule that's secreted out, and now it's going to go and have a response. Well, the extracellular signaling molecules that are secreted, can either bind to cell surface receptors, right? Which is the typical thing that we think about, that it's coming in, it's binding to a receptor, it's changing the receptor and causing the signaling cascade. But they can actually also be internalized and bind to a receptor inside the cell, either in the cytoplasm or even in the nuclear membrane. So it could be at any different level. An example of that would be retinoic acid, which is a very potent differentiation factor. It doesn't bind to any receptor on the plasma membrane, but actually gets into the membrane, through the membrane into the cytoplasm, and binds to the retinoic acid receptors inside. Your dexamethasone experiment that you looked at in your lab also had similar stuff, where dexamethasone didn't bind to a receptor on the surface. It actually went through the plasma membrane and bound to dexamethasone receptors inside the cell. So there are many molecules that actually get internalized into the cell and then bind to their receptors inside the cytoplasm or even nuclear membrane, and then elicit a response, usually by controlling gene expression in the nucleus itself. On the cell surface receptors, many times these molecules will bind to their receptors, where they cause a conformation change or activation or suppression of that receptor that then leads to intracellular signaling cascade in return. Okay. Now, depending upon how this molecule binds and what type of steps it has to take, the response of the cell can be very fast, or in milliseconds even, or could be a lot slower process, which takes minutes or hours or days even. So an extracellular signaling molecule, where all it needs to do is alter, a particular signaling cascade, just alter a protein to get it activated or inactivated, will lead to a very fast response, right? It just does it, it activates it or suppresses it, and that's it. The cell is different. However, a cell surface signaling molecule, where it needs to activate a whole set of proteins that then have to regulate gene expression and lead to then new mRNAs getting formed and then new proteins getting synthesized, will take time. that's going to take minutes to hours or even days. So the altered cell behavior can be looked at as a way to guess, as molecular biologists, whether you expect the signaling molecule to be affecting gene expression or not. So if you're just using a molecule for the first time, we could look at how the cell changes and how quickly it responds to see if it is an intracellular signaling pathway or something that requires alter protein expression. So something that's going to relate to gene expression regulation. Okay? Up till there? That's a fine. Okay, so then this is something that's also talked about in that lecture that I posted. There are steps to these signaling cascades that are predetermined, that are going to be consistent in every single signaling pathway. You should be able to recognize these several, various parts of a signaling pathway. So we always start off with obviously a signal, whatever signal it is, whether it's a molecule or a UV radiation zapping the cell, whatever it is. That signaling molecule will bind to some receptor in many cases, either extracellularly or internally. And then it will lead to some mediators. These mediators are your intracellular signaling molecules that will usually amplify the signal through each step. And then that is going to lead to effector proteins downstream of it, which will do one of three functions. It will either alter a metabolic enzyme to do its work that it needs to do and change the level of metabolism in the cell. It could be a cytoskeletal protein that is getting affected that may change the shape or movement ability of the cell, or it may be a transcriptional regulator, in which case it's going to alter gene expression at the end of the day and change the way which proteins are getting expressed by that cell. So these are three main things that you can see as a response at the end of the day from the effector proteins that are activated by your signaling molecule. So let's look at what that means. This kind of system, is what we would call a primary transduction, where the portion of your pathway where the signal is getting bound to the receptor, right? That's your original signal, and activating a mediator protein. So this is your primary transduction. All these intracellular signaling molecules, they will be transducing the signal as well as amplifying it. So that's your amplification step of your pathway. Then the effector proteins that are made in response to the signal are your feedback proteins. These proteins will then control the response. That's the final act that the signaling pathway is leading to, whether it is life, death, differentiation, growth, division, whatever it is. That final response is going to be affected by these effector proteins in the pathway. Okay? Yes? Right, 5 kids. How are we in time? Good. Okay. So now so this was again a signaling molecule that's getting secreted, whether it is through endocrine signaling or paracrine, but something that is secreted and then goes to the target cells and then elicits a response. Now let's look at a cell that is a contact dependent signaling control. So here is an example how that may happen, right? So in this example, you have these unspecified epithelial cells that get differentiated and develop into their final types of cells, final types of developed epithelial cells based on the signaling that they receive. That signal is received by the main nerve cell developing form from the epithelial cells. And this is going to produce that starting signal that's going to be at its plasma membrane. So all the cells, all the unspecified epithelial cells, kind of these partially differentiated epithelial cells that are in physical contact with that cell membrane and are bound to that signal through their receptors, will then cause them to develop into the way that they need to develop, whether they are going to be inhibited in their function or increased in their function. So that kind of targets them to then stop growing further and dividing out and making more of these epithelial cells, and allows them to specialize into the cells that can control the nerve cell that's right there. Now, the proteins that are in this case responsible are the delta proteins, which is your membrane-bound inhibitory signal protein to inhibit these epithelial cells from reproducing and dividing and growing more. And then the notch protein is the receptor protein that gets expressed on these epithelial cells as they become the ones that are integrated with that nerve cell and to help its function work. Okay. So the central cell was also once upon a time a epithelial cell, but this got differentiated into that nerve cell, and all the cells that are going to be around it are going to then form that helper structure to maintain its function the correct way. Now, this job is done, like we said, through many different types of signals. A lot of our signals in our body are actually either endocrine or paracrine, so they are secreted and not membrane bound like the ones that you just saw. That's a minority of the signals in many cases. Many small hydrophobic hormones can simply bind to intracellular receptors and act as transcriptional regulators because they can readily move through the plasma membrane because they're hydrophobic, they're small, and they can just go through and affect the cell inside. So this includes a bunch of our hormones like the cortisol, estradiol, testosterone, all of the main ones that we are looking at every day. So when that happens in that case, again, the same general pathway is going to be seen, right? So whether the extracellular signaling molecule is binding to the receptor or it's going into the cell and then binding to its intracellular messenger, it's going to still have a primary transduction step, an amplification step, and a feedback step. So this is, again, another example of the same method that I just showed you earlier. In this case, you see the amplification of the signal through release of a second messenger. So this is where you can think about that secondary response in a way. So your primary signal was up here that's bound to the receptor. The first set of relay of the signals led to an activation of a protein that then produces a second small intracellular messenger molecule. This secondary messenger molecule is going to be used to amplify that signal. So that one molecule is now leading to multiple secondary messengers getting produced that then bind to their receptors that can then cause the feedback system to make your altered gene expression or to make the response that it needs to make. Now, many times this system has a feedback loop to either positively regulate it or negatively regulate it to further amplify or to stop the signaling cascade as needed. And whether it amplifies it or stops it, inhibits it, depends upon the incoming signal and the type of response that you're looking at the end of the day. Okay. So here is, again, example of those positive and negative feedback regulation. In that step, once you get, once you get up here, right, when you have that secondary messenger and now it's amplified the signal and it's having a response, that response will lead to different proteins getting produced or getting activated or inhibited. When that response needs to be amplified further, then that secondary response is going to go back and reactivate that intermediate step and continue that loop until it's stopped by another protein. In a negative feedback, however, once that secondary messenger has elicited its response, the final protein would come back and inhibit that intermediate step to adjust that extracellular signal and stop it from continuing on any further. So in this case, once this final protein is made that's going to alter the gene expression, it is also going to then go back and inhibit this protein. One of the proteins that it produces will inhibit that intermediate step to stop that signal from continuing any further. Okay? Do you guys get everyone good here?
Most of the time, is it like more negative feedback in our body, or it depends?
I think it depends. I'm not sure if I know if I can for sure say that it's mostly negative, because there is a lot of positive feedbacks as well. I could look it up and tell you, but either way, both of these are very important in our everyday life, and both of them are happening all the time. So the intracellular signals are gonna get, again, we'll actually learn about that in a little bit more. So this extracellular signaling molecule, let's say that is your molecule that is binding. As long as it's able to relay the signal, it will cause this transduction and amplification step. Now, some signaling molecules, they will only activate once. And once they activate this protein, they kind of fall off, or they are unable to activate that receptor anymore for a certain amount of time before they can reactivate it. And that's, again, an internal regulatory mechanism. Other molecules, they can stay bound and stay, as long as they're bound, this is active, this is happening. So it depends on the molecule that is binding and the receptor that they are binding to, whether it will be a continued signal or whether it will be a one-time signal and then that's it. It cannot do it for a set amount of time again, unless there's a new molecule that comes and binds. Okay.
Okay, thank you.
What happened?
I think both of us said thank you.
Oh, okay. You're welcome. Okay. So the intracellular signaling proteins, basically, these secondary messengers in a way, the intermediate proteins, as well as the signaling proteins that come in, like those small hormones that get into the cell to activate signals. Both of those can act as molecular switches. So, one level of activation is on the cell membrane itself, but this is an internal molecular switch mechanism. So one example of this is where a signaling molecule comes in and then activates by binding to a protein kinase, activates it so that it gets phosphorylated through ATP, right? And that phosphorylated signal is then that is activated, and that can do the work that it needs to do. That can respond in a particular, cause the cell to respond in a particular way. Another way is through activation of G proteins or GTP binding proteins. And that's a different type of, it's similar, but different. In this case, the normal signaling proteins, GTP binding proteins are bound with a GTP molecule. So they already have a diphosphate on them. And the coming signal swaps it by taking the GDP off and causing a GTP to come in and bind to the same protein, causing it to get activated, where then it can do its work. So in the first case, it was not swapping a molecule. In this case, the signaling molecule was activating the protein through activation of protein kinase, which then phosphorylated the protein of interest that could then do the work. And then in the other case, in the GTP binding proteins, the GTP protein itself is bound with a GDP that the signaling molecule is going to swap out with a GTP to activate it itself. So this has an intermediate step, this one doesn't. Or intermediate, both of them have, but intermediator, this one has a mediator protein, this one doesn't. And that signal then can do the work and get you the final work. So this is again looking at how the GTP binding protein is activated by these signaling molecules. So normally you have your proteins that are present with a bound GTP, and those are not going to be functional until you know a new exchange factor that is activated swaps that GDP with a GTP. So it's taking a GTP molecule off, and in its place, it's putting in a GTP that then activates that protein and can now do its work. Now, these activated proteins then can also work with gap proteins, which are GTPase activating protein, that will then dephosphorylate it and bring it back to its inactive state. So again, this is a way to regulate the signaling pathway so that it doesn't go haywire. You know, so think of a signaling molecule that may be a growth molecule, right? A growth hormone. If it was never switched off, it was no regulatory switch to make sure that there was not too much activation, then it will continue to make this again and again and again, continue the cell to grow and divide, grow and divide. And that's what may lead to cancer or another issue. So that's why it's important to have multiple levels of regulation throughout the cell signaling cascade. Yes, Taylor.
Which one had the mediator protein? Was it the signaling by protein phosphorylation?
So yes, the ATP, using ATP to phosphorylate a protein through a protein kinase has the mediator.
Okay, cool. Thank you.
Okay, so now cell surface receptors, again, these are the ones that all the signaling molecules are going to get bound to. They fall into many different classes. So they can, depending upon what type of receptor you have, you will have different options of responses. Some of them are going to be ion channels coupled receptors. So that's an example with that skeletal muscle that we were looking at, right? So the channel is normally closed unless acetylcholine binds to it. When it opens up, that would be an example of it. And then you have an influx of ions that will really lead to the muscle contraction response, right? So that's an example of an ion channel coupled receptor. Other receptors are what we call G protein coupled receptors. These are the ones that are going to utilize these GTP models, the GTPase and the guanine nucleotide exchange factors, to regulate their activation. And that usually is a 3-protein complex. So it includes a receptor, an inactive or an active G protein, depending on what type of G protein coupled receptor it is, but basically a G protein and some enzyme that they are going to then affect based on their activation level. So that's your G protein coupled receptor. And then you would have enzyme coupled receptors that must be bound with some kind of an enzyme that they are activating at the end of the day, right? Or they can be the enzyme themselves with some catalytic site on them. So in this case, you can see an example of an enzyme coupled receptor where the signaling molecule is joining in as a dimer, bringing those two proteins together to form an active catalytic domain that can then do the work. Or you can have a single signaling molecule that comes to bind it to activate the receptor that then can activate the associated enzyme to do its function, to do the catalytic activity it needs to do. So that would be another example of that. G protein coupled receptors are by far the ones that have the most complex cascades of reaction. They have many, many, many steps that they go through to amplify the signals, to regulate themselves, to go through the feedback loops of all types. They usually are the more complex signaling systems. Okay. Okay, so I have examples of some extracellular, these are the ones from your book, extracellular ligands that can bind to these receptors. And they are going to give you a little bit information about whether they are ion channel or G protein coupled. If I have a question on the exam for it, and I do, I'm telling you each time if it is, I'm in most cases, I'm telling you if it's a G protein or an ion channel coupled receptor. So it's not something that you would need to memorize as such, but you should kind of look at it to see if you can find some, similarities that you can kind of remember. So you see these molecules, right? The nicotine, morphine, heroin, capsicum, menthol, all of these small molecules that mimic particular types of activation neurotransmitter signals, right? Activating or inhibitory neurotransmitter signals, and then you can look at what type of response they would cause, and be kind of able to bring it together and synthesize that to explain how they would have activated the particular channel that they activate. Okay. Here are some hormones. Again, you don't need to as such know too much details about them. You should know that these are hormones, right? And you don't need to know necessarily the site of origin for them. For this class, you would probably learn that in anatomy and physiology anyway. But for now, you just need to know the hormones and what type of thing they're derived from. So most of these are going to be derived from cholesterols. There are only two in here that are just protein based, and that's the adrenaline, the epinephrine, and the insulin. All the others require cholesterol as part of their structure. And then here are examples of some other signaling molecules that are present that are, specifically either local mediators or neurotransmitter or content dependent. Again, and when I'm giving these to you inside a question, I'm usually telling you, especially for critical analysis, I'm usually going to say it's a local mediator versus a contact dependent signaling molecule. How is the process, how is the signaling pathway different for these two? So you should be able to explain that, right? Okay, so we're going to talk about, for the last few minutes, we are going to talk about G protein coupled receptors, okay? So G protein coupled receptors, these are going to be bound into the plasma membrane. And many times they are multi-pass transmembrane proteins with a binding site for the signaling molecule on the extracellular side. These proteins, this is the typical structure of them. They'll have 7, transmembrane spaces throughout their molecule. The extracellular space is specifically for the signaling molecule to bind. The intracellular side of that protein is where the other proteins are going to be the proteins that interact with that G protein coupled receptor are going to be housed. They can be bound to it actually in real life, but be inactive, or they can be nearby, depending on what receptor it is that we are looking at. But the actual these 3 proteins, they're always the same. This is the inactive G protein. It has 3 subunits, alpha, beta, and gamma, that are bound with the GDP molecule. This, like I said, will be housed in the cytoplasmic side of the plasma membrane, integrated into the plasma membrane on that side. It's not transmembrane, and it's integrated using a lipid linker. Now, this is going to interact with the receptor protein in order to cause the response. Now, in some cases, the signaling molecule comes in and activates the G protein, which allows this to come bind to it. And in that case, once it's bound to the activated receptor, the conformation of this molecule changes, causing GDP to be dissociated, which allows GDP to get bound to it. The binding of GDP causes the subunits, the activated subunits, to get separated and elicit the response. So activation of this leads to binding of this to, binding activation of the G proteins, where GDP gets dissociated. not activation, full activation, but just GTP dissociation, which then allows for GTP to get bound to the to the G protein to activate it, which can then cause the signaling pathway and response. Now, once it is activated, now it has to figure out what it needs to do. And that's going to depend on what type of G protein it is and what type of final function it needs to perform. Many times there'll be a target protein nearby that it's going to bind to, that it will then activate in response to it, or elicit a response through it. Now, this is usually done just by the alpha subunit, and the beta and the gamma complex, once activated, will kind of separate out from the alpha subunit. The alpha subunit with the GTP that's bound to it will activate its target protein, which will then cause it to lose the GTP to GDP because it dephosphorylates it. And then that allows the beta and the gamma subunit to come back and rebind to the system to form that 3-unit complex, which is the inactive G protein. So the active G protein is actually separated from the alpha subunit, from the beta and gamma subunit. And then once this GTP is used up, is hydrolyzed, you will then rejoin those three to make the inactive G protein again. Okay? Yes? No? What is the G protein cycle? What it accomplishes in the cell is it accomplishes activation of a target protein that will then activate downstream signaling pathway. So this is the first step to the response. What it allows for a cell to do is to use a small number of G protein receptors and a small number of these G proteins to activate a host of different types of responses by controlling which molecules which target protein they're going to activate downstream. Okay. So some G proteins are obviously actively, once activate, remain bind to the membrane. They don't ever leave the membrane. And while they're there, they will activate their target protein, which will then cause the second messengers to be formed and diffused to act as intracellular signaling pathways, molecules. So this is the second messengers that are coming because of this activation. In other cases, your proteins may actually activate another protein on the plasma membrane itself. So it's not doing an intracellular response, but rather it is activating a channel or another protein on the plasma membrane. In this case, the beta and the gamma subunits, once activated, are binding to a potassium channel, causing it to open and releasing the potassium out into the space, right? So both things can happen. It doesn't always have to be that the GTP, G protein, when activated, is only going to cause intracellular signaling cascade. Sometimes it's binding to a receptor on or another protein on the plasma membrane and activating it, many times regulating ion channels through its activation. Another thing that some G proteins use as an intermediate second messenger signaling molecule is using either inositol triphosphate, IP3, or diacylglycerol molecules as second messengers. So once they are activated, they are going to activate a target protein that will release or that will cause these IP3 and DHG molecules to be activated or released into the cytosol, where they can then activate downstream signaling pathway. Okay, so here is an example where the first activated signal molecule that activates the G protein. The G protein, once activated, then leads to activation of a phospholipase C, which causes the inositol phospholipid to release these IP3 molecules that can then bind to their own activated, their own receptors. It could be in the ER lumen that they are binding to a receptor to open calcium channels and releasing calcium, or they could be binding to something in the plasma membrane itself. So that's another way that they can work through amplification of signals. So G protein, basically the G protein cycle allows a method for amplification of signal through use of second messengers that they activate. Okay, so we're going to actually stop after this. Fertilization of an egg, and we talked about that last time a little bit, that I was going to show you something cool with that. By a sperm triggers a G protein activation that leads to increase in cytosolic calcium ion concentration in the egg that prevents it from getting fertilized again by another sperm, right? It kind of calls it home at that time. And you can see that happening in this video. If I can click on it. Hopefully it will open it so I can show you, and then I will go. Too many times. So what you see is that at the moment of fertilization, you will see this, spark essentially happening on these cells as they are fertilized and as they kind of secure their boundaries so that no other sperm can get in. Oof, no, I don't want to. It has really weird music, so I'm not going to play the music. But these flashes of light is because of that calcium ion concentration getting increased at the moment of fertilization. And what they have noted is that, especially, they noted this in vitro fertilization cases, and they found out that these are, again, zinc is the ion that's produced later on, but it's calcium and zinc production. They believe that the eggs that have the brighter flash at fertilization are actually healthier embryos later on. And those are the ones that go further into development and are without any issues and can be used very successfully. So that's as far as we got, and that's as far as you will have on your test. Everything else is not going to be on there. So we will finish that up next time. Question?